Bismillahirrahmanirrahim. Today we will discuss about introduction of bioinformatics, main databases used in bioinformatics, and aim and scope of bioinformatics. So first we will study a simple statement by Professor Donald, that biology easily has 500 years of exciting problems to work on. From last 100 years, from last 1,000 years, or from 500 years, we have millions of problems related to biology, and we have to solve that, including diseases and other different types of problems related to bioinformatics and biology. And bulk amount of data was generated from human genome projects and other biological programs, and it was a big job. It was a big job to analyze all the data, to store all the data, and to interpret all the data that we have. So what are the main, first we will discuss about the main collaborative domains of bioinformatics. Usually bioinformatics just touch three major domains, including genomics, proteomics, and transcriptomics. And first we will have a look to all three basic domains, and after that we will jump on to the other sections. A living cell, a living cell is a system where cellular components such as genome, the gene transcript, and proteins interact with each other, and these interactions determine the facts of the cell, whether a stem cell is going to become a liver cell or a brain cell or a heart cell. So in genomics, we just play with the genome of any organism, maybe the genome of human or a genome of any other organism, including animals, plants. So genomics has a significant role in all the biological research, because we cannot move without genome study, without nucleotides, the base of genome in actual, the chromosomes, the genes, and the location of genes, where it in actual lies, how it works, what it will be the expression level of those proteins, sorry, those genes and nucleic acids. So all these study is included in genomics, and the extensive analysis and insight from that specific gene or genome can made with the help of bioinformatics and molecular biology techniques. It is a science that attempts to describe a living organisms in terms of the sequence of its genome. I don't know why this window is appearing again and again. Anyhow, genomics uses the technique of molecular biology, cell biology, biochemistry, and bioinformatics to explore in actual what genome has, what that specific genome of that specific organism has, how these sequences works, how these sequences have similarities, where is the coding sequences, where are the non-coding sequences, how we can regulate the different genes, and how these genes work. So the first automatic DNA sequencer was developed in 1986 by Laurie Hood, and influenza virus was the first bacterium to be sequenced in 1995 with the help of genomics. Now we have a simple introduction of transcriptomics. Transcriptomics in actual is the study of complete set of messenger RNA and the molecule of messenger RNA that has a specific function and involvement in the biological population cells. This study will help us to analyze or study the different expression levels of genes, and different techniques are available that can help us to study the expression level of genes, including DNA microarray analysis. And these kinds of advanced techniques help the researchers to dig more about genome and other related studies to monitor and to regulate the expression of different genes for the treatment of different diseases. In proteomics, in actual, we study about the proteome and proteins. What are the structure of proteins, three-dimensional structures, motifs, domains, how these structures behave, how problems happen to the structures, and how we can fix the problems, and sequencing of proteins that will be the amino acids, and these all are included in proteomics. And bioinformatics help proteomics to analyze all these data related to structures of protein. If we cannot have the funds, or we cannot have the time or expertise in X-ray crystallography and NMR, we can jump on bioinformatics approaches and techniques to solve the three-dimensional structure of a specific novel protein. Different metabolic proteins are also interpreted and analyzed by bioinformatics approaches and techniques. So researchers feel very easy with the help of bioinformatics approaches to solve the proteomics problems. Now we have genomics and proteomics together, where genomic is the study of an organism genome and the use of genes. It deals with the systematic use of genome information associated with other data to provide answers in biology, medicine, and industry. However, in proteomics, we just study the proteome and proteins of that specific organism at a wide level, including the function of that proteome and proteins, and the structure of the protein and proteome, and what will be the defected structure of that proteins. Proteomics is much more complicated than genomics, as in proteomics we have post-translational modifications and different types of proteins with the help of different types of forms because of RNA shuffling and RNA exon shuffling and RNA splicing, and we have more proteins as compared to genes. So in gene, we just have to study about the gene and expression of the gene in genomics. However, in proteomics, we have to study about the three-dimensional structures, interactions, how these interactions happen, in which environment these instructions happen, and what will happen if the environment will change. So proteomics is considered more complex as compared to genomics. So nowadays we have different databases available in the field of proteomics and genomics with the help of bioinformatics. And day by day we have an increased number of databases related to proteomics and genomics data. So here we have a modern map makers that map the entire human genome here. And we know that the human genome is of 3.2 billion base pairs, but we cannot interpret these 3.2 billion base pairs without the help of bioinformatics. We have different types of metabolisms in our body, and there are so many metabolic pathways, and a researcher cannot understand fully or completely any metabolic activity or pathway without bioinformatics. So bioinformatics is nowadays yet everywhere. Here we have some figure that if we have this, how we can measure the activity or the process of gene activity, and how we can measure the metabolic processes. So here we have some diagram with the help of bioinformatics that how these genes has a relationship with each other and interaction with each other with the help of protein interaction and protein ligand interaction. Recently, we have different gene activity measurements. However, in 1989, the first microarray technique was discovered by Professor Fodor. The photo is here. So he just invent this approach of microarray analysis. With that microarray analysis, we can study more than one gene at a time, and the expression of gene and different properties and characteristics of gene with the help of microarray analysis. In genomics, we have DNA, and everyone knows well about the DNA who is related or the student of bioinformatics or biology. DNA is a nucleic acid that contains the genetic instructions specifying the biological development of all cellular forms of life. DNA is a long polymer of nucleotides and encodes the sequence of the amino acid residues in proteins using the genetic code, a triplet code of nucleotide. So here we have different examples of cells, eukaryotic cells, prokaryotic cells, and what these cells in actual have, how post-translational modifications occur, and these all are the processes we studied in past with the help of genomics and proteomics. Here we have a diagram of DNA under electron microscope, and here we have the three-dimensional structure of DNA with the help of bioinformatics. So Watson and Crick, in 1953, they just give the three-dimensional structure of DNA, and how this what the DNA in actual has, which databases, purines, pyrimidines, and which was the double ring, and which has single ring, how this works, where we have the pentose sugar, where we have the ribose sugar. So this all is just to recall the biology what we have. So when we have the base once again regarding genomics and proteomics, then we can fully understand what bioinformatics is and where bioinformatics help us. So the genetic codes, as you guys know, the three nucleotides just comes together and code one amino acid. The genetic code is a set of rules that maps DNA sequence to proteins in the living cell and is employed in the process of protein synthesis. Almost all living things use the same genetic code called the standard genetic code. Although a few organisms use minor variations of the standard code. So in proteomics, we have primary structure that is the linear amino acid sequence of a protein. The main structure, the key structure of all the amino acids are same, like has a side chain and then has a main chain that will be constant. However, the side chain has variables and change depends on the structure and nature of the amino acids. So here we have secondary structure that includes alpha helices, beta plated sheets, or three-dimensional structure that will be supercoiled, and there will be a binding pocket interacting site that will function. So after that, we have another domain, that is evolutionary domain, or you can say evolutionary biology. Nowadays, many researchers are working on computational evolutionary biology and generated so many trees that can help us to understand the evolutionary biology and the comparison and similarities between the genomes compared to genomic studies. So now, after recalling our genomics, proteomics, transcriptomics, and other basic biology concepts regarding protein, DNA, and different pathways, we will jump on. Now we are jumping on bioinformatics. What is the definition of bioinformatics? Simple. We have the simple definition of bioinformatics, that any computational approach that can help to solve any biological problem is in actual bioinformatics. So this is the most simplest definition we have. In actual, bioinformatics is the advanced interdisciplinary science of biology that contains biological problems, mathematical formulas, and concepts, and computational power. Bioinformatics developed new thoughts for the researchers to maintain the different concepts, maintain the different concepts and data analyzed, and have to analyze the data and store that huge amount of data in different databases, so other researchers can retrieve it easily. In actual, bioinformatics is a logical and technical approach by which not only we can solve the different biological problems, but we can also give different predictions related to biological problems. The first look, if we will have to the bioinformatics, then we will jump on genomics. In actual or sequence analysis, how we have to analyze different genome sequences. A mathematical model should be, here we have a statement by Einstein that mathematical model should be simple as possible, but not too simple. But in this statement, the researcher just want to explain that we should have to generate so simple algorithms and models to solve the problems, so we can have a conclusion or logical conclusions for a specific problems. In bioinformatics, we have another definition of bioinformatics. Bioinformatics is the science concerned with the development and application of computer hardware and software to the acquisition, storage, analysis, and visualization of biological information. It has the following three concepts in actual, that how we can develop different advanced new algorithms with the help of statistical formulas and approaches and ideas to solve the biological problems, how we can store all the data and where we have to store it, and how we can retrieve it again, and how we have to annotate this data, and what will be the logical conclusion we have to drive with this data. Application of bioinformatics tools are just to analyze all the data we have, the interpretation of all the data with the help of different bioinformatics tools. The development of databases should be very efficient, user friendly, accessible for everyone, and the data should be managed sequentially. So everyone can easily understand all the data and interpret the data, use the data for different experiments. Bioinformatics drives knowledge from computer analysis of biological data. These databases consist of information stored in genetic code, experimental results from various sources, patients, statistics, and scientific literature. Different bioinformatics research have different types of domains, including storing of data. That is the most important job by bioinformaticians, to store the data in meaningful way and then have to retrieve the data and analyze the data. Bioinformatics is growing day by day and helping researchers to solve different biological problems, because it is an interdisciplinary science and have all the concepts and techniques of computer sciences, mathematics, statistics, chemistry, physics, and all the related domains. It has many practical applications in different areas of biology and medicine to solve different problems of biology. Bioinformatics is the application of information technology to store, organize, and analyze the vast amount of biological data generated by different researchers. The stored data should be available in a sequenced form so everyone can retrieve the data of proteins or amino acids for their own research work. The biological information of nucleic acids is available in numerous databases, and we can easily retrieve the protein and amino acid data from that databases. Sequences are represented in single dimension. However, structures have three-dimensional structures. Three-dimensional. So here we have the basic domains of bioinformatics. From this one, bioinformatics, we just can go towards computational languages. For with these computational languages, we can generate tools. We can produce new databases. We can generate tools. We can generate softwares for solving the biological problems of genomics and proteomics. And we have biostatistics, and we have R language in this, so we can solve different problems of proteomics with the help of that. So after that, we have computational biology, chem informatics, database management system, and system biology. All these are the basic areas of bioinformatics to solve different biological problems. In computational biology, we have structure prediction with the help of homology modeling, threading, ab initio, phylogenetic analysis, computational evolutionary biology, protein structure prediction, evaluation, protein three-dimensional binding pocket analysis, and how we can interpret the interacting residues, protein docking analysis, protein ligand docking analysis, visual screening, and simulation of different protein structures. So what is the history of bioinformatics in actual? The collection of amino acid sequences was compiled in the Atlas of Protein Sequence and Structure by National Biomedical Foundation. This collection was edited by Dayhoff from 1965 to 1968, almost 50 years back. Dayhoff and his colleagues just contribute to the comparison of amino acid sequence by developing different effective computer softwares, so they can easily detect the relationship between the sequences. And in 1980, the EMBL, European Molecular Biology Laboratory, was established, and the EMBL established different data libraries to collect all the data from all over the world related to nucleic acid and proteins. NCBI was established in USA, and it is considered as the primary information data bank that has all the data from different researchers, and they store the data in one database, so we can easily extract that data or retrieve that data for our experiments. After that, the National Biomedical Research Foundation was established in 1984. In 1859, we have, if we will jump back, first we have Charles Darwin introduced us with the genomics and evolutionary biology. So after that, we have different databases and different analysis related to these sequences. In 1869, the DNA from nuclei of white blood cells was first isolated by Frederick. In 1951, Pauling and Corey proposed the structure of alpha helix and beta pleated sheets that we will study in three-dimensional structure of proteins. In 1953, as you guys know, Watson and Crick just proposed the double helix structure of DNA based on the X-ray data of Roshan and Franklin. In 1955, the sequence of the first protein was predicted and analyzed by Professor Sanger. In 1958, the Advanced Research Project Agency was formulated by USA, so everyone just can have all the data. In 1973, PDB was announced, and nowadays PDB Protein Data Bank is one of the most finest database for protein structures. In 1987, Perl language was generated by Laurie Wall, and with the help of this language, we can just generate so many softwares and tools to solve the biological problems. In 1988, National Center for Biotechnology Information was founded. In 1990, Human Genome Project just launched, and everyone was working on the researchers were working on Human Genome Project, and BLAST was introduced at that time by Carlin and Archer, and this is one of the finest software nowadays we use for different comparison and local searches. So BLAST is one of the best search tool we have now till 2021. In 1992, the Institute for Genome Research Associated with claims to exploit sequencing commercially through gene identification and drug discovery was found. In 2001, Human Genome Project was completed, and we found 3.2 billion base pairs of data related to human genome. That is a big, that time that was a big problem, that how we can interpret and how we can store and retrieve this data. In 1958, Professor Sanger just got the Nobel Prize by developing a protein sequencing technique that is one of the finest technique we have nowadays also. In 1978, Professor Sanger just completed the first viral genome. In 1980, first mitochondrial genome was sequenced by Professor Sanger. In 1980, he again got the Nobel Prize for developing the DNA sequencing technique. In 1995, complete genome of influenza virus was published. In 2001, entire genome of Homo sapien was published, as we know that. So in genomic area, we have different organism that was sequenced in 1978. We have the sequence of this first viral genome in 1980, first organelle genome that was human mitochondrial genome. 1985, we have AIDS, retroviral genome, HIV. In 1995, we have first bacterial genome, and in 2001, we have the complete human genome project sequence. So what was the future goals of molecular biology and bioinformatics in actual till 2050? Researchers just want to develop a computational model of a complete cell. So in a cell, so many organelles we have, and every organelle has a specific function and just floating in cytoplasm. So researchers are trying to generate a computational model of cell, that complete cell, that how every organelle is working, so we can study and simulate this data. So Human Genome Project was the you can say landmark or hallmark of bioinformatics. Then that time we have bulk amount of data related to human genome, and to analyze that data, to understand that data was a big problem. So US government just started a project with the help of Department of Energy and the National Institute of Health in 1986. And genome was in actual, the hydrating material of any organism. So at that time, what was the main aim of the Human Genome Project was to just analyze all the genes of human DNA, and how these genes works, and how many database, how many base pairs of genome we have, how we can store this data, where we have to store this data, how we can interpret this data, and how we can retrieve this data. So all this was the basic aim of Human Genome Project that time, and we got much benefit from that Human Genome Project. The key benefit we have in actual was drug discovery and personalized medicine. And with the help of Human Genome Project, we can cope out or overcome so many diseases, improvement in medicine and drugs, and we can develop new drugs with the help of the available data, microbial genome research for biofuel and environmental cleanup, DNA fingerprinting and forensics, improved agriculture sciences, a better understanding of species related to evolutionary biology with the help of Human Genome Project, and also to study the gene mapping with less risk assessments. Internet and mathematics has, you know, very strong relationship. Without internet, we cannot retrieve the data. We cannot study the bioinformatics well. Internet plays a key role to retrieve the biological data that we have in different databases. The computational part of bioinformatics used to optimize the biological problems, and here we have computer science. And with the help of computer science in bioinformatics, we can generate different databases. We can develop databases. We can generate different operating systems. We can generate different tools and softwares to solve the biological problems. The mathematical portion of bioinformatics help the researcher to understand the algorithms designed for solving a specific biological problem and also to generate different bioinformatics tools. The mathematical portion of bioinformatics has different matrices, biostatistical analysis, integration, differential equations, complex mathematical functions, transformations. So after with these concepts, we just incorporate these concepts to generate new algorithms to solve the biological problems. Internet is a global data communication system. With the help of internet, we can retrieve data from all over the world, and we have different softwares and hardware structures and infrastructures that provide connectivity between the computers to retrieve the data. The web is one of the services communicated via the internet. It is a collection of interconnected documents and other resources linked by hyperlink and URLs, as you know guys well about that. So internet resources for bioinformatics, we have different databases, search engine, as we studied that NCBI was generated, EMBL was generated about 20-30 years back, so we can have data in that one. After that, we should need a specific search engine from where we can retrieve this data. Without retrieving this data will be of no use for us. So database search engine, we have different Uniprot, NCBI, DDBJ, Swissprot, online servers. We have ClusterW for different multiple sequence alignment for database management systems. We have to generate different databases. We have different softwares and information sources to produce new softwares. Online databases has KEGG for metabolic analysis, Modbase for three-dimensional structure predictions, PDB for protein structures, zinc database that has different compounds available, natural compounds and other chemical compounds. We have different types of biological databases that are primary, secondary, composite databases, bioinformatics software that help us to solve the biological problems. So these are the resources of bioinformatics. We have bioinformatics databases, biological databases, different types of biological databases, including bibliographic databases that has all the data about information, published literature information, taxonomic databases, nucleic acid databases that has the data of nucleic acid and DNA, taxonomic databases has the classification of all the available data. Genomic databases has the genome data and gene level information of different organisms. Protein databases has the three-dimensional structures, protein information, interaction sites and locations. Protein family domains that has all the data about different protein families, relationship between the families and the different proteins. Enzyme and metabolic databases has different metabolic pathways, how these pathways works and the relationship of these pathways with each other. So we have online databases, GenBank, EMBL, DDBJ, DNA Data Bank of Japan. FASTA is standard data format usually used for different databases for searching and retrieving. So in generalized form, we have databases of DNA, proteins, carbohydrates, metabolic pathways, three-dimensional structures, protein family databases, EST tags, GST tags, SNPs databases, genome databases, microarray analysis databases. So different millions of databases are available that has different types of data. Overview of databases. Databases indexing and specification of search terms to retrieve, follow up, and analyze the available data. In archives, we have databases of nucleic acid sequence, genomes, protein sequence structures, proteomics, expression pathways, different types of database. Gateway of archives includes that how we can jump into that archives, what are the search engines we have. NCBI enters, PubMed, XPSC, Swissport, and etc. Generalized DNA, protein, and carbohydrate databases includes primary sequence databases like EMBL, European Molecular Biology, laboratory nucleotide sequence database at EBI. GenBank is also one of the famous database by NCBI, and DDBJ is the DNA data bank of Japan that has all the data about DNA. Different types of biological databases accessibility for different sequence analysis. So we have, as we already discussed, that we have primary databases, secondary databases, and composite databases. Primary databases in actual has all the data directly come from different researchers and different laboratories. In secondary databases, we have curated data. However, in composite databases, we have the combination of data from primary databases and secondary databases. Here we have primary nucleic acid and protein databases. In nucleic acid primary databases, we have EMBL, GenBank, DDBJ. For protein, we have PIR, Tremble, and SwissProt. For secondary databases, we have ProCyte, PFARM, Prints, and Blocks. These are these databases to interpret the protein family, domains, and different interaction sites. In composite databases, we have the combination of primary database source like NRDB and OWL. You guys have to Google these databases and have a look to NRDB and OWL. These are the most informative databases we have in actual. So the international sequences database collection is like around here. We can see that the data from EMBL just move and have pipelines to also show in GenBank, from GenBank to DDBJ, and from DDBJ to EMBL. So in short, if we have data in one database, we just have cross references that we can jump into other database for the related data. GenBank is the NIH genetic sequence database and annotated collection of all publicly available DNA sequences. The DNA Data Bank of Japan is the DNA database initiated by Japan in 1986 at the National Institute of Genetics. So the Center of Information Biology at NIG was reorganized as the Center for Information Biology and DNA Data Bank of Japan in 2001. The new center is to play a major role in carrying out research in information biology and to run DDBJ operation in the whole world. So EMBL nucleotide sequence database constitutes European primary nucleotide sequence resource, main source of RNA and DNA data sequences, and the researchers can directly submit the DNA or RNA sequences to these databases for different work and different analysis. The database is produced in an international collaboration with GenBank and the DNA Data Bank of Japan. So NCBI, we have National Center for Biotechnology Information that was established in 1988 as a resource of molecular biology information, and this publicly available database has different data related to computational biology to develop different softwares to analyze the analyze the data with the help of softwares and tools, and all for the better understanding of molecular processes affecting the human health and different diseases. EBI is the European Bioinformatics Institution that is a non-profitable academic organization that help and generated with the help of European Molecular Biology Laboratory EMBL. EBI is a center for research and services in bioinformatics. The institute manages databases of biological data, including nucleic acid, protein sequences, and macromolecular structures. DDBJ DNA Data Bank of Japan is established in 1986 with the help of Japan and the NIG and Ministry of Japan. So in this database, all the nucleotide sequences and all the different databases related to nucleic acid sequences are connected with DDBJ. DNA sequences records the organismic evolution more directly than other biological materials, thus invaluable not only for research in life sciences but also human welfare in general. The database are, so to speak, a common trigger for human beings. So here we have SwissProt, XPacy proteomics server that help to analyze the proteomics data, and all the proteomics data is available related to bioinformatics on XPacy. You can easily retrieve the data and analyze the data that you want to analyze. Generalized DNA, protein, and carbohydrate databases, we have SwissProt, Tremble, PIR, PSD, UniProt, UniRef, IPI. So these are all the databases, carbohydrate databases. And in carbohydrate databases, we have ArcBank, three-dimensional database, PDB, EBI, MSD, and DB. These all databases have different data related to genes, nucleic acid sequences, and protein sequences. And if you are interested, you just have to Google the name of these databases and you can have a look to all the databases. So every database has different search engines. Without search engine, there is no need of that database. So the main concept, we have data in databases and we have to treat the data with the help of different search engines. So here we have the search engine of Enter, that is text based, sequence based for BLAST and FASTA, motive based for scan, cross site and emotive, structure based for structure similar search, and mass based for protein search and different protein analysis. So what was the initial goal of bioinformatics at that time? To acquire the data, store the data, retrieve the data, analyze the data, develop different tools to analyze that data. Data acquisition is primarily concerned with accessing and storing of data generated by different researchers and biological experiments. The data generated by different projects related to protein sequences and nucleic acid sequences and retrieval of those sequences are appreciable and capable of being linked to all information related to proteins and DNA samples. The data should be organized in different databases for easy retrieval, so researchers can easily retrieve all the data and just analyze the data with the help of bioinformatics tools. Biological information should not be hidden for everyone. It should be available for everyone with the help of internet and with the help of databases, different databases. It should use for betterment. It should use for to improve the future molecular biology, future molecular medicine, future cell biology, and different future approaches. Tools and databases, the database development is the core concept of bioinformatics at that time, because different approaches like in genomics, proteomics, and different experimental biology approaches just generating a bulk amount of data. However, many people don't know how to interpret that data, where to store that data, and how to retrieve that data. That was in actual the main job of bioinformatics. Data analysis, to use these tools to analyze the data and interpret the results in a biologically meaningful manner. Efficient analysis requires an efficiently designed databases. Data integration, once information has been analyzed, a researcher must often associate or integrate it with the related data from other databases. So we have molecular medicine to improve the the medicine, generate new medicines and develop new medicine related to that specific gene and protein. When we have that all the data, the Human Genome Project help us to improve our molecular medicine related to every disorder, every disease, including cancer. So this may be inherited or the result of the body response to an environmental stress can also be improved with the help of available data or just doing some mutations and alteration in the genome. The Human Genome Project help us to analyze all the genes directly associated with different diseases and begin to understand the molecular basis of these diseases more clearly. uh, we have another application as personalized medicine. In personalized medicine, we just sequence the genome of a specific person or individual and generate the specific and develop the specific medicine and drugs for that specific individual. So drug development, at present, all drugs on the market are just related to some available proteins of 500 or 1000 proteins. However, with the help of bioinformatics and computational tools, we can specifically improve our drug development, and we should be more specific to that specific gene, target specific drugs for specific disease. Gene therapy is the approach used to treat, cure, or even prevent disease by changing the expression of a person's genes. Currently, this field is in its infantile stage with clinical trials for many different types of cancer and other diseases ongoing. So what was the basic need of bioinformatics at that time and nowadays even? So the data available from Human Genome Project, how to retrieve, again and again we are revising the same concept, that where to store, how to retrieve, where to use, how to interpret with, and how to understand the biology in actual with the help of bioinformatics. There are major fields of bioinformatics: molecular medicine, gene therapy, drug development. We discussed all. So bioinformatics has participation in molecular biology, biochemistry, cell biology, protein expression level, gene expression level, genomic, proteomics, simulation, and molecular docking analysis. Protein structure prediction is the main one of the main key application of bioinformatics. With the help of bioinformatics, we can predict the secondary, tertiary, and quaternary structure of different proteins for different analysis. We can annotate the genome, and we can regulate different sequences and genes with the help of bioinformatics. Comparative genomics, we can study the structure and function of two different or more than two different genomes with the help of bioinformatics. We can improve the health and drug discovery and development processes with the help of bioinformatics to improve the health conditions in human beings. Other applications include waste cleanup, microbial genome applications, antibiotic resistance, alternative energy sources, crop improvement, development of resistant varieties, forensic analysis, bioweapon creation, insect resistance, and sequence analysis. In actual, we just study what bioinformatics is. Bioinformatics is in actual the understanding of biological problem, solving of biological problem with the help of computer power and mathematical and statistical approaches and formulas. To solve biological problem with the help of computation power is known as bioinformatics in actual. There are bioinformatics helps after human genome projects, genomics and proteomics to understand everything, and to understand genome data, to understand nucleic acid data, to understand protein data, proteomics data, three-dimensional structures, microarray analysis, and bioinformatics has different bioinformatics has applications in different domains and fields of biology. So in actual and in short, bioinformatics help us a lot. Every biologist needs bioinformatics nowadays. Thank you so much, everyone.